Hello, my name is Joshua Mugabi. I welcome you to Topic 1, Cell Biology, Learning Outcome C. Analyze the ultrastructure of animal/plant cells, bacterial cells, and the plasma membrane to distinguish prokaryotic and eukaryotic cell characteristics. We are looking at learning activity A. In groups, learners search, study, and compare images of ultrastructures of animals or plant cells with a bacterium cell and make diagrams using a suitable graphic program. Study images of the fluid mosaic model of the plasma membrane. Make a model and use it to explain the functions of the membrane and its part. Make a write-up and present the findings to the class. Comparing ultrastructure of eukaryotic and prokaryotic. Item 1. We shall be comparing cell ultrastructures. Students are provided with electronic micrographs of plant cells, animal cells, and a bacterial cell. They are required to study the images, compare the ultrastructures, and produce clear diagrams. Task: identify 3 organelles common to both plants and animal cells. Identify 2 structures present in plant cells but absent in animal cells. State 3 features present in bacterial cells but absent in eukaryotic cells. Compare the nuclear material of eukaryotic and bacterial cells. State 4 guidelines for producing biological diagrams. Identify 3 organelles common to both plants and animal cells. Of the nucleus, mitochondria, endoplasmic reticulum, or Golgi apparatus. Identify 2 structures present in plant cells but absent in animal cells. We have the cell wall, chloroplasts, large permanent vacuole. State 3 features present in the bacterial cells but absent in eukaryotic cells. No true nucleus, nucleoid DNA, no membrane-bound organelles, presence of plasmids, peptidoglycan cell wall. Compare the nuclear material of eukaryotic and bacterial cells. Eukaryotic cells, DNA enclosed in a nuclear membrane as chromosomes. Bacterial cells, DNA free in the cytoplasm as a cellular molecule. State 4 guidelines for producing biological diagrams. Use clear, unbroken lines. Label structures neatly using straight label lines. Do not shade or color. Diagrams must be large and proportional. Include a title. Now let us look at comparing ultrastructure of eukaryotic and prokaryotic. Item 2. We shall look at the functional significance of cell differences. A learner asks why antibiotics can kill bacteria without affecting human cells. Task: explain the reason using structure difference. Identify one bacterial structure targeted by antibiotics. Explain the reason using structure differences. Bacteria lack membrane-bound organelles found in human cells. Antibiotics target structures unique to bacteria. Identify one bacterial structure targeted by antibiotics. Cell wall, peptidoglycan, and ribosome 70S. Let's now look at fluid mosaic model of the plasma membrane. Item 3. Studying the fluid mosaic model. Students study an image of the fluid mosaic model of the plasma membrane and are asked to construct a physical or digital model to explain membrane structure and function. Task: describe the basic structure of the plasma membrane. Name 4 components of the plasma membrane. Explain why the membrane is described as fluid, mosaic. State 2 functions of each of the following: phospholipids, proteins, carbohydrates. Describe the basic structure of the plasma membrane. The plasma membrane consists of a phospholipid bilayer with embedded proteins. Name 4 components of the plasma membrane. Phospholipids, proteins, carbohydrates, and cholesterol for animal cells. Explain why the membrane is described as fluid, mosaic. Fluid because it contains phospholipids that can move laterally. Mosaic, proteins are scattered within the bilayer. State 2 functions of each of the following phospholipids: proteins, carbohydrates. Phospholipids, they form a semi-permeable barrier and maintain the membrane structure. Proteins transport substances across the membrane and act as enzyme or receptors. Carbohydrates, they are for cell recognition and cell adhesion. Fluid mosaic model of the plasma membrane, item 4. We shall look at using the model to explain membrane function. A student uses a membrane model to explain how glucose enters a cell. Task: identify the membrane components involved. Name the transport mechanism. Explain how membrane structure supports this process. Identify the membrane component involved. Carrier protein. Name the transport mechanism. Facilitated diffusion. Explain how membrane structure supports this process. The protein spans the bilayer. It binds glucose and changes shape to move it across. Selective permeability is maintained. Expectations summary. Correctly identify ultrastructures. Make accurate comparisons. Produce neat labeled diagrams. Correctly explain membrane structure and function. Apply knowledge to real biological situations. Conclusion. Structural differences between cells explain their functions, while the fluid mosaic structure of the plasma membrane enables selective transport and cell communication. Thank you for participating in Topic 1 Cell Biology. We have been looking at learning outcome C. Analyze the ultrastructure of animal and plant cells, bacterial cells, and the plasma membrane to distinguish prokaryotic and eukaryotic cell characteristics. We have been looking at learning activity A, where in groups learner study and compare images of ultrastructures of animal or plant cells with a bacterium cell and make diagrams using a suitable graphic program. They study the images of the fluid mosaic model of the plasma membrane and make a model and use it to explain the functions of the membrane and its part. Make a write-up and present the finding to the class. If the following has been useful, visit me at Mugabi Biology on YouTube.